All right, this is your review for the final exam for quarter 4. So pay attention, knuckleheads. All right, so the first thing is this is about DNA. Remember that DNA is your genetic material. And we use our DNA for a lot of things. Our DNA is what gives us, the traits that we have and everything. Your DNA is found in the nucleus of all of your cells. And remember that your DNA is like 2 billion nucleotides long. Okay. Remember that a nucleotide is composed of 3 parts. It's a sugar, a phosphate, and a nitrogen base. Okay, so if you look at this picture of DNA right here, this is a sugar, this is a phosphate, this is a base, so this whole thing is a nucleotide right here. Okay, so our DNA is made up of billions of nucleotides long that make up our DNA strand. Okay, remember that DNA is complementary. It's 2 strands that are complementary to each other. And so that just means that the opposite side of the DNA strand has that opposite nitrogen base to it. Okay, so looking at this picture, this is your backbone right here. This is pointing to guanine and adenine and cytosine and thymine, which are nitrogen bases. This is pointing to a covalent bond. So remember that there's a strong covalent bond here that holds together the sugars and phosphates in the backbone. And that this is an ionic bond right here, letter E, and that holds the nitrogen bases to A, the complements. Remember that our complementary base pairs are A to T and C to G. Okay? So remember that DNA basically is found in different forms and different varieties, being the same thing. Remember that a gene is a segment of DNA. Okay, so that's a segment of DNA. So, this is all pointing to genes. These different segments, like from here to here, is a gene. And from here to here is a gene. It's just, a segment of DNA, a chunk of DNA. Okay, remember that your DNA coils up to form a chromosome. Okay, so remember that your DNA forms up to form your chromosomes. Remember that the chromosomes are only formed during mitosis, really during replication of mitosis and when you're starting to divide your cells. So normally your DNA is just DNA in your cells, and then it only forms chromosomes to reproduce. So we form chromosomes just to pass on DNA from cell to cell. Okay, otherwise it's just spaghetti noodles. All right, here's another chunk of DNA, another gene here, and this is showing you the complementary base pairs A to T and C to G once again. So we talked about some different parts to chromosomes. So remember that each arm of a chromosome is called a chromatid. And that the middle part that holds it together is called the centromere. And remember that these are called sister chromatids. And they are sister chromatids because one is a copy of the other. And after replication, they copy each other and they form X's during mitosis. Okay, remember that the tips of the chromosome are called the telomeres. And remember, that was the cap that just keeps it from unfraying. Lastly, remember that all of these little bands that you see on here are actual genes that are found on that. So speaking of those genes over here, remember that there was 2 different kinds of genes. There was what we called exons, and there was introns. And so this is just a quick way of showing you that an intron is an in-between non-coding sequence of DNA, whereas the exons are coding chunks of DNA, and they code for a protein. So this whole thing is a gene. And this again is pointing to that telomere. Okay, lastly, just showing you the base pairs here. Make sure that you can do this. I know that this is a chunk of DNA because I've got the second strand here and I've got T's in it. So put the complementary pair. This is AAT, ATA, TAA, and TGC. And then 3 and 5. Okay, the next topic is about DNA replication. So remember that DNA replication is when we copy our DNA. Right, so that's making a copy of your DNA. So DNA replication happens in your cells to your DNA when you need to reproduce. Happens before mitosis. Okay, it happens before mitosis. So the process was fairly straightforward. Remember, you've got a chunk of DNA and you've got to basically unzip that DNA, and that was the job of an enzyme called helicase. So this unzips the DNA, and so that's happening right here. Okay, we're unzipping the DNA so that we can get at the information. Okay, then remember that the enzymes called DNA polymerase are making new DNA. Okay, and so we're making new DNA. And remember that that's made in opposite directions according to the strand. So this one you can see is being made toward the replication fork, and this one's being made away. If it goes toward the fork, we call that the leading strand. And remember that DNA is made from 5' to 3' if the numbers are given to you. In this case, they're not given. So this is the leading strand, and remember that this has a DNA polymerase that is going this way and making DNA. This chunk's being made away, so this is called the lagging strand, and it's being made in the wrong direction. So that has several enzymes that are making DNA in this direction. Remember that they're made in small chunks called Okazaki fragments. Okay, and those are the little chunks that are made in the lagging strand. Okay, so that's DNA replication. Remember that in human replication, our DNA replicates really slowly because our DNA is pretty big. We can replicate our DNA in 6 hours. In bacteria, since bacterial DNA is circular, it can replicate itself in 20 minutes. Very, very small amount of time. Okay. Remember also that we make mistakes in our DNA when we make our copies, and those are mutations that happen, just the wrong letter gets put, and DNA polymerase also has a job of proofreading for mistakes, and it catches all but 1 billion of its mistakes, and those are permanent. Okay, the next topic is the cell cycle. Remember, the cell cycle is basically just what is the cell doing during its lifetime. So this is a cell lifetime. Okay, and so there was 4 different components to it. So I'll start in G1 with a brand new baby cell. And so G1, remember, is for growth and a cell doing its job. Okay, so cells are growing, they're doing their job, and they're just, doing the things they need to do. Okay, at some point they're going to go through the S phase, which is called synthesis, but the S phase really means DNA replication. So this is that stage of mitosis of the cell cycle where replication is happening, and so we're copying our DNA, and so it's not doing its job during this. Then we're going to go to the G2 stage, and that's more growth, and that's more doing its job. And that's really getting ready, preparation for mitosis. Okay, and that's what the M stage is, that's mitosis. And so remember, that's where we're doing our stages of mitosis, which is really PMAT, prophase, metaphase, anaphase, telophase. Remember that all of this, G1, S, and G2, are all just interphase. Okay? Okay, so just looking at the results of the 2 different processes of mitosis and meiosis, The cells here on the left are from mitosis. And what that's showing is that when you start with a cell in mitosis, you start with an average everyday cell, which is a diploid cell. We use the letters 2N to represent diploid or normal. So this could be a skin cell or anything like that. When it does mitosis, we produce two cells that are identical to each other and identical to their parent, and they are also diploid cells. So we start with a skin cell, we get two skin cells, and they're both the same. Okay? That's not the case when we do meiosis. So remember, meiosis, we're trying to get cells that are not diploid anymore. So we start with an average everyday cell, again a normal diploid cell, but then what happens is it's going to divide in half. Remember the first part of meiosis, we split it in half. So they become 1N. And then what they do is they go through that second stage and they split all apart, and you take the sticks apart and they continue to be N haploid cells. So these are all haploid. They all have half the genetic material, and these are sperm or eggs. Okay? And so they are genetically different from each other and genetically different from their parent, whereas these are the same identical cells. Okay, stop. Get out of the picture, please. You're distracting me. Okay, so as far as replication and everything goes, we talked a little bit about mutations. Remember, mutations are just changes of DNA. They're just any change to your DNA that is going to affect you. It affects you because it messes up the message that your DNA makes, which means ultimately it changes the protein that is produced, and it is different. And so you're producing a different protein than you were before. Okay, so different ways that we get mutations. Number one, we get mistakes during replication. Remember that DNA polymerase makes 1 in a million mistakes, and they don't all get fixed. So they makes a lot of mistakes putting the wrong letter. Okay, so these are normal mistakes to your DNA, and they don't really affect you. Remember, your cells reproduce about 60 times in their life, and when they're done, and those cells die off. Furthermore, when you affect that, you make one skin cell have a mutation that didn't happen before, it's not really a big deal. These are called somatic cells. And so somatic cell is every cell in your body. So if you just affect a skin cell, no big deal. Mutations to it really aren't a big deal. And you can't pass them on to your offspring. You don't pass a mutation to a skin cell off to your offspring. Now, if it's a mutation during meiosis, in that copy process, that's a lot worse because these are sex cells. And so if you have a mutation to a sex cell, you are for sure going to pass that off onto your offspring. That's called heritable. And so you're going to pass it on your offspring. So that's a lot worse. We talked about mistakes during meiosis, like Down syndrome and those different disorders, and those are passed on to your offspring. Okay, so we talked about external factors like radiation and chemicals, getting x-rays, things like that, that can cause mutations to your DNA, even sunlight. Okay, if you're exposed to the sunlight, that causes, cancer, skin cancer. Okay. All right, we also did karyotyping. We looked at different karyotypes. Remember that a karyotype is just a listing of your chromosomes. Okay, so this is a listing of chromosomes. So remember that we have 23 pairs or 46 chromosomes, and that's a normal adult individual. If it doesn't have 23 or 46, then that is a mutation. It's caused by a thing called non-disjunction, and that's again how you get like Down syndrome. So we did karyotyping. We got these and we counted to make sure. We found in a lot of the cases that there were mistakes. For example, if there was 3 number 21s right here, that was Down syndrome. And if there was XXY, that was called Klinefelter's. And if there was X0 and there was nothing here, that was called Turner's syndrome. So there was some really specific examples of where the meiosis didn't work, you didn't get the right chromosomes. Okay. We know that looking at the karyotype, that we can determine the gender of an individual based on its karyotype. So remember that boys are XY and girls are XX. So this is a boy right here. This is a male because he has a Y right there. So Y's determine gender. XX is girl, XY is boy. Okay? So that's karyotyping. I'd expect to see a couple of karyotypes on there with a mutation. Okay, and from there we talked about what fertilization was. Fertilization is basically when the sperm hits the egg. So here's how it's happening in real life here. And this is something called in vitro fertilization. And so this might be where we fertilize an egg in a test tube or a petri dish. Okay, and that happens now, especially with all the biotechnology and cloning. They'll fertilize an egg in a petri dish like this, and then they'll still grow that baby in a surrogate mother. So either way, whether it's au naturel down here or in a test tube here, both of them, fertilization happens up in the fallopian tubes up in here. And then the fertilized egg, these are called gametes. Remember that gametes are sex cells that are unfertilized. They're haploid cells. They get fertilized, they travel down the fallopian tubes, and they're going to go into the uterus in here, where they're going to embed on a wall, and that's where they're going to grow. And that's going to take about 7 days to get there, and then they're going to grow for about 40 weeks. And so fertilization happens in the fallopian tubes out here. It could be a surrogate mother. It doesn't even have to be the right mom. For example, here, this test tube, you could fertilize it here and stick it in an elephant, theoretically, and stick it in the uterus. As long as you can get it to hook to the wall and get blood supply, it'll grow, and it would be a human. So that's pretty crazy. Remember, I talked about a specific group of cells called a blastocyst. This is a 4-day embryo here. And it's got this hollow ball of cells in the middle here, and these were special because these were embryonic stem cells. And those are important because they can turn into any kind of cell in the human body. And so, that's where cloning and things like that are going to happen in the future. And then once it goes from a blastocyst and it continues to grow, it's going to grow from an embryo into a fetus, and it's going to grow for 40 weeks and turn into a big, bouncing, healthy, hopefully, baby. Okay, next is mitosis. Mitosis is cell division. So remember that mitosis, we start with chromosomes. Remember those chromosomes in interphase will replicate and copy, so I'll make them as X's. Okay, so this is interphase. In after replication, the nuclear envelope breaks down, and these things are random X's. In metaphase, they start to line up in the middle, so you get this like this. In anaphase, they start to split apart. And so they're elongating, they're getting pulled apart, and eventually that pinches apart, and you get these cells that are here. And those cells, the nuclear envelope reforms, and they go back to being an everyday cell. This is a diploid cell to begin with, and these are diploid cells to end with. Remember that these are not X's anymore, because once they're done, they go back to being just spaghetti noodles. So these only X'd after replication. Okay, meiosis is how sex cells are produced. So we start with a diploid cell, and again, it's going to start as spaghetti noodles, and then after that, it's going to replicate and form X's. But remember, these X's are random pairs of X's, and then they'll line up in the middle as pairs in metaphase, and then anaphase, they actually pull apart, and so now we've got our 4 X's. So we've already divided our cells into haploid cells. We split them in half right down the middle here. And these are the homologous pairs. These are the copies of each other. Because they started as this, and then they turned into that. Okay, then these chromosomes are gonna line up as X's here, just like in mitosis, and then they're gonna split apart, and like that. And so now we have haploid sex cells that become sperm or eggs, and they are not identical to each other. So it has a lot more steps to it, but remember the whole second half is really just mitosis. It's the exact same thing as mitosis. Okay, next is transcription. Transcription is when we take our DNA and we make, this is the process of making proteins, but this first part is taking your DNA and making a mRNA copy of it. So we're copying it as messenger RNA. Remember, this is happening in the nucleus. So our DNA is in there. We unzip our DNA just like before, and we have a new enzyme. This one's called RNA polymerase. And so RNA polymerase is going to go through, it's going to copy this DNA strand, and it's going to make what we call a messenger RNA copy. That messenger RNA copy is going to leave the nucleus, and it's going to go out into the cytoplasm. Here you can see that messenger RNA strand. I know it's messenger RNA and not DNA because A, it is single-stranded, whereas DNA is double-stranded, and B, I can see that there's U's here instead of T's, so there's no T's in here. There's U's instead. Okay, so it follows the same base pair rules. A goes to U and C goes to G. So if you have like TAT, CGC, that becomes AUA, GCG. So once the messenger RNA strand is in the cytoplasm, it's going to do a thing called translation. Translation is going to be how we copy the RNA into proteins. Okay, so what it's going to do is that messenger RNA strand is going to go out, it's going to find a ribosome, because ribosomes are where protein synthesis happens. And so they're going to go out into the cytoplasm and find a ribosome. Remember, they've got these funny looking little tRNA things. And so the tRNA has their own little anticodon on it. They're going to find the codon like AUG, and they're going to spit out the complement, and they're actually going to spit out these little amino acids. And so remember that amino acids are what are made in translation, and then they're later stuck together by a bond, and that's what becomes our protein. So translation is copying our RNA into amino acids, which it makes into proteins. Okay, so lastly, then you should be able to use this chart to figure out what protein you made. So if I give you a DNA, let's say it's TAC, you've got to first translate that into messenger RNA, which would be AUG. You're going to notice that AUG is usually first, and AUG, if you find it, U and G, so that's going to be right here, that's going to be MET. So that kicks out MET as the amino acid, and that would become your protein. So your protein is just those amino acids all hooked together. Okay, so if I give you one more, let's say it is CGA, then that becomes GCU, and then that becomes GCU. It becomes ALA. So remember, you only put the first three letters. You'll have this chart on the test, and it'll just be multiple choice. Notice there's one start codon, and there are 3 stop codons. When you reach a stop codon, it just tells it to stop. You've made the right protein. So back to our mutation stuff. If you make a mutation to your DNA, it changes the DNA sequence, which then changes the messenger RNA, which then changes your protein and jacks up your whole body.
Okay, next was Punnett squares. Punnett squares is a way of looking at genetics and figuring out what possible offspring you would have due to those offspring. So the first example I gave you was a boy and a girl. So remember that boys are X, Y, and girls are X, X. So if you do the Punnett square, two of these individuals are going to be girls, and two of these individuals are going to be boys. And what this means is this is probability. That means if they had 4 kids, they have a 50% chance that 2 of them would be this and 2 would be this. So if they have any kids, they've got a 50-50 chance that it would be a boy or a girl. So we would write this as a ratio of 2xy to 2xx, and that would be my genotypic ratio. Remember, the genotypic ratio is what letters it has. Or my phenotypic ratio, which would be 2 boys to 2 girls. And so that means I have 50% probability. Okay, so a couple examples. I had Tall Brian and Short Dakota, just random names off the top of my head. Tall Brian is heterozygous, so he's big T, little t. So heterozygous means big, little, like big T, big R, little r. It means you've got a mixture of the letters. They are opposites. And you have Dakota, who is short, so she's little t. We refer to her as recessive, which is the lowercase letter. And she's homozygous. Homo's the same, so those are the 2 smaller letters. So little t, little s, whatever. Okay, dominant is the capital letters. And if there's a dominant trait there, that is the trait that is seen because it dominates the recessive. So look at our cross between Brian and Dakota. We have big T, little t, big T, little t, little t, little t, and little t, little t. So my genotypic ratio is 2 big T, little t to 2 little t. What letters does it have for genes? Or my phenotypic ratio, what does it look like physically? And that is 2 tall to 2 short. Will any of these individuals be medium height? No, there is no such thing as medium when we're talking about these kinds of genetics, okay? So don't let me trick you. Okay, the last one was Smooth Sydney and Wrinkly Wyatt. Smooth Sydney, I believe, was heterozygous, so that would make her big S, little s. And Wyatt was wrinkly, so that was little s, little s. So that means that that smooth is dominant to wrinkly. And so big S is smooth and little s is wrinkled. Okay, put these together, you get the same ratio. Oops, that's little s. As before, you get a 2 to 2 ratio. So my genotypic ratio is 2 big S, little s to 2 little s, little s. And my phenotypic ratio is 2 smooth to 2 wrinkled. Okay, so expect to see some Punnett squares on there. Okay, in the home stretch, let's talk about the worms. So the worm was one of the first land creatures that we looked at. The worm was significant because it was the first organism with an enclosed digestive system. Okay, so we talked a lot about the significant things that worms do. You know, that they breathe through their skin, and they have to stay moist, and that they eat dirt, and they poop dirt, and that they were what we call invertebrates. So they have no backbone. Okay, some of the distinguishing features about worms. Up here is a brain, so it did have a brain, wasn't very big, but a brain nonetheless. This is the esophagus. Might be called the pharynx, same thing. Okay, These two little guys right here were the seminal receptacles. So those are the seminal receptacles. That's where it's going to receive semen from another worm. These right here were the 5 aortic arches, which is the worm's hearts. Okay, and then these were the seminal vesicles. So that is the equivalent of the worm's testes. Okay, so we know that worms are what we call hermaphrodites because they contain both male and female sex organs. Okay, almost done. Here's the crop and the gizzard. And the crop and gizzard are used to hold, store, and grind up food, and its food is dirt. And then lastly, this is just the enclosed intestine. Okay, so these are eggs for the worm, and here's the worm's mouth. All right, these up here are pointing to the segments of the worm. That's what makes a worm an annelid. And this was called the clitellum, which we know is a sexual organ to help hold them together. And this is the anus. So that's worms. Okay, fish. So we looked at fish a little bit different. Fish was different because they're not a terrestrial animal, they're an aquatic organism. And fish were different because they had a backbone, which makes them a vertebrate. And fish have lots of advanced features for living in the water. They have gills to get oxygen out of. That's this number right here, 24. They have a swim bladder inside them, which is number 25. And that's used to obtain water to move the fish up and down. We talked about the couple different things about the fish, that it has a 2-chambered heart, called an aorta and a ventricle, and that's this right here, 22, and that it has a more advanced digestive system. It's more advanced than the worms. Okay, so basically the fish eats the food. It goes through the esophagus, it goes into the stomach here, right here, number 19, and then it's going to go down into the pyloric cecum, into the intestine. It's going to go down the intestine, and it's going to wind out, and it's going to go down the anus here at number 9. So a much more advanced digestive system because it's eating more advanced foods. Okay, over here was the gonad. We saw eggs in the females and testes in the males. This is number 17. And again, here's the swim bladder right here. Otherwise, we saw the liver and kidneys were up in here, and the heart right here. And here's the gills. And it did have nostrils way up here at number 3. And we talked about the different fins briefly. This is the dorsal fin, and this is the anal fin, and this is the pelvic fin, and this is the caudal fin. That's what it's used to swim with. Okay, so that's the fish. And last but not least, the frog. The frog was the first animal we looked at that is both terrestrial and aquatic, so it has kind of characteristics of both things. So externally, we looked at the frog, and we talked a lot about its camouflage and the fact that it has ears called tympanic membranes. And that's the ear. And we talked about the fact that its tongue was backwards facing, and that it had nostrils and its eyes were on top. We talked a lot about the skin and everything about that. So that was the external stuff. The frog has one anus on top, and that's where it does all its business. We also talked about the difference in the front feet and the back and the webbing. So internally, the frog was pretty cool. We looked inside the frog. We found that it had an esophagus where the food goes down. It's going to go into the stomach right here, and it's going to go down. It's going to go into the small intestine and then into the large intestine. Here's the small intestine, and here's the large intestine right here. Any of these 3 could be called the large intestine. I'd live with it. This is probably the the cloaca, and this is probably the anus, but we're splitting hairs. They're all part of the large intestine. So point being is that the frog has a more advanced digestive system still. It's got a small intestine, which should do most of the chemical digestion of the food, and then it has a large intestine, which basically just stores poop. Other organs we looked at the gallbladder right here. The gallbladder stores bile, which is a digestive juice. We saw the kidneys were in the back of the frog. And we saw the pancreas was right here. And it was right here between the small intestine and the stomach. We also saw the spleen down here. The spleen is a blood reservoir. And this is probably just a part of the small intestine. I wouldn't worry about it. Okay. Lastly, we saw the lungs were up here. I don't know if that's what these things are pointing to. We'll just call it as such. And we know that the frog has large lungs because it holds its breath for long periods of time. Okay. So the frog is definitely well advanced for living in water or on land. Okay. Frogs breed sexually, as do fish, as do worms, but they all just do it just a little bit differently. So I think that's everything. Hope you do great on your exam. Hope this helped you very much. And good luck. Okay, peace. Thanks, buddy.